Thank you. I'm going to introduce Jouni Uitto, who is the head of our center in Pennsylvania, and specifically Philadelphia, who has worked with us a very long time. Many of you are familiar with Jouni's work, and I'm really delighted to have him speak to us about genetics and anything else he thinks we should know. Thanks, Jouni.
Sure, pleasure to. Let me see if we get the share business going here. Can anybody see my screen now?
Yes. Yep. Looks great.
Okay.
Perfect.
And okay, let me move these faces out of the way. So yes, afternoon, sunny afternoon here in Philadelphia, the city of brotherly love. And as some say, sisterly affection. So greetings to everybody. Thanks, Sharon, for inviting me to give a little bit overview of genetics in PXE. And this is all done, or most of the work is done under the auspices of PXE International Center of Excellence that is sponsored by PXE International here in Department of Dermatology and Cutaneous Biology at Thomas Jefferson University. And most of the work, especially all the preclinical animal work that I'm going to talk about, is done by Kwali Lee, or Lily, who's been 15 years now my collaborator here in Jefferson. So let's start a little scenario here. You discuss with your neighbor and you say, wow, my child was diagnosed with PXE. And the neighbor, unless the neighbor is a professor of genetics or dermatology, she may say, what is PXE? What is that? Well, you say PXE is a syndromic, late onset, fully penetrant genetic disorder with autosomal recessive inheritance, with genotypic heterogeneity and phenotypic intra- and interfamilial variability with prospects of allelic therapy. So what do you say now? You say, what is this? Okay, basically, this lecture that I'm giving, it's sort of structured to be a genetics of PXE 101. So hopefully you all will be, towards the end of this lecture, able to describe what all these fancy terms mean. So just come back to what Sharon mentioned. PXE actually was described initially about 100 years ago. It was a French dermatologist, Balzea, who described the skin lesions in 1884. And then there was another French dermatologist, Darrier, who coined the term pseudoxanthome élastique to describe these skin findings, 1896. But then the connection of the skin findings and the eye findings was actually described by two Swedish doctors, a dermatologist and an ophthalmologist, who in the late 1920s make this connection. So it's a syndromic disease. In other words, you have at least two different organs affected. So, and in fact, it's eponymic syndrome. Eponym means that it's called Grönblatt-Strandberg syndrome, as in earlier literature describing PXE. Well, from the historical perspective, Jouni Uitto published his first paper in PXE in 1971, well before many of you were born. And Sharon Terry actually established the PXE International in 1995. And the gene defect in ABCC6 gene was disclosed in 2000. So those are the highlights of the history of PXE. So even though I published the first paper in 1971, I really got seriously interested in PXE in 1995 when Sharon came to my office here in Philadelphia, telling that her daughter, Elizabeth, shown here, and Ian, the younger son, were diagnosed a year earlier by Leon Berkovich as PXE. And Sharon obviously went to the books and nothing was known about the disease. The genetics was unclear. The overall history was not very clear. What is going to be, how severe the disease, what's happening. So Sharon said, Dr. Uitto, you have to help us. And I said, sure, happy to help, but why don't we clone the gene? Sharon asked, well, what needs to be this? And I said, well, we need 400 families, their DNA. It's going to do a little work. And she thanked, left, and quit her day job and established the PXE International. This was 1995. She came back about a year later, says, well, here is the DNA from 400 families. Let's find a gene. And in collaboration with the PXE International, we and others established a consortium of 4 laboratories which worked together very hard using state-of-the-art techniques at that time, but it still took close to about 2 years to find the gene. But make the long story short, we eventually reported in 2000 that the mutated gene in PXE is ABCC6, which, if I get this pointer working here, no, it isn't.
Jouni, can you move? There's a little thing that says, please move this window away, and it keeps coming on and off. I don't know what it is. It's toward the top of the slide. Do you see that, or do we just
I
see
see
that?
on the background there, yeah.
What is this? I don't know what that means. I've never seen that before.
Move this window away, no.
But it doesn't move. Okay, it's okay. It's most of the time it's not covering anything. All right.
Well, let me see if I can move it. No.
No, that's really weird. Okay, and it fades for a while and then it comes back. I don't know what the deal is.
Please move.
Okay.
I don't know. Okay.
We'll solve PXE, but not slides. Go ahead.
Okay. Don't know. I haven't seen that before, but I just was telling Tamara my computer was upgraded to Windows 10 and we had all kinds of problems. So
yeah.
Nevertheless, so we identified ABCC6 as the gene that harbors mutations in PXE. And the ABCC6 actually stands for ATP binding cassette family C member 6. It's a protein that is on the surface of the cells. And it was predicted to pump out something out of the cells to the extracellular matrix. And that somehow then results in normal situation, prevention of calcification. But if this pump doesn't work because of the mutations in this gene, then we get the disease, PXE, which is a calcification disorder. So we very quickly, and yeah, this is annoying.
No, it's
well,
really weird.
I can change to the other computer, but let's see how this is going.
Sure.
So we very quickly identified mutations in this ABCC6 gene in a large number of families, and these are the series of the 100 first families that we identified in PXE patients. And this is basically a structure of the entire PXE, ABCC6 gene, 31 different coding regions, and all these arrows point to the mutations, so-called nonsense mutations in this gene, which means that the kind of synthesis of the protein stops where this arrowhead is. So in most cases, you don't, or any of these cases, you don't make a full-length protein. So it's a non-functional, so-called loss of function protein. And that obviously causes the non-functional protein to being present and causes the disease. We also had so-called MISSENSE mutations. That means that you make the whole full-length protein, but you have these critical amino acid substitutions in the protein in different parts of the molecule. And they particularly concentrate in this part, which is sort of critical for the function of this pump as a functional protein. So this was the first 100 mutations. So now we know it's a genetic disease. In other words, there are changes in the DNA. But how is it inherited in the families? Is it autosomal dominant? Which basically means that one mutant gene copy. So you inherit one copy from the mother and one copy from the father. And these copies are called alleles. So if you have one copy affected, and that causes the disease that's called autosomal dominant form of the disease. If both copies of the ABCC6 in this case are mutated, then you get an autosomal recessive disease, and the parents who are heterozygous have only one of the copies affected. They are perfectly normal. So this is autosomal recessive inheritance. And then there is an X-linked form of the disease, but that's predominantly in the males. And that wouldn't be applicable here because we have pretty much equal numbers. They may be a little bit more females, but in general, pretty much females and males having a PXE. And also this implies that the mutated gene is in an X chromosome. Our gene is chromosome number 16 in the short arm. So this is out. But is it autosomal dominant or autosomal recessive? Before we studied this gene cloning and mutation analysis, there were suggestions of both kind of mutations or mode of inheritance, and you cannot clinically tell, so you need the mutation analysis. And actually, this is from the very first paper we published, and these are here 4 different families with PXE. And those who are not familiar with the pedigrees, so all these black symbols, they are PXE patients. And the open symbols are clinically unaffected individuals. And of course, you know, the squares are males. Males are usually pretty square guys. The round ones are females. They are a little rounder, a little softer. So these are females, males. And we had here families. You look, for example, here, this family, the parents are clinically normal, has 4 affected daughters and 1 unaffected son. Here is another family, 2 clinically affected parents, although unaffected parents, although the father was dead, but the 2 affected daughters and their children are not affected with PXE. And similar situation with this family 1. So that is all consistent with autosomal recessive inheritance. But then somebody points out here, how about this one? Here is mother to daughter inheritance. And actually this family was genetically counseled that it's autosomal dominant disease, which would imply that this daughter's children all have a 1 in 2 chance of having PXE. However, if we look now into the mutations, turns out that actually this daughter had inherited 1 mutation from the mother, but she had this has also inherited a mutation from the heterozygous father, who was related distantly through this family loop in the consanguineous family to the mother. So this patient also has autosomal recessive disease. And the implication is that her risk of having an affected child, instead of being 1 in 2, is something order 1 in the 5,000. So we clearly have a autosomal recessive inheritance, which basically means that the heterozygous carriers are clinically normal. That's the parents in most cases, and some siblings. And the risk of these 2 heterozygous parents to have an affected child in each pregnancy is 1 in 4 having an affected with PXE in both alleles. So 25% of the chance of the children in each case. Heterozygous carriers, the chance is 50%, 2 out of 4. so that the one allele is mutated, but they are clinically normal. And then 1 in 4 or 25%, they have only normal alleles, so no mutations in their ABCC6 genes at all. And this kind of mode of inheritance, it's fully penetrant. That means that if you have inherited 2 copies of the mutant gene, all these affected individuals will eventually develop PXE in essentially 100% of the cases, while the heterozygous carriers do not develop clinical findings at all. Now, we did some recent calculations of carrier frequency. In other words, how many of these heterozygous people are there in the United States? And we came in this number 1 in 2,275. And if you do the calculation, there are about 340 million people in the United States. So if you calculate, there is about 150,000 carriers who are clinically normal, but have one of their ABCC6 alleles mutated. So there is always a risk of these carriers meeting and having children, and those children then each have the 1 in 4 or 25% chance of having the PXE. Now, you don't burn the odds up. So every child has this 25% chance. If you have 5 children coming with PXE, the 5th one still has the 25% chance. So you didn't make any assumptions based on previous history. Now, phenotypic heterogeneity, what do we need for that? One of the examples is that PXE is really a late onset. It's a fully penetrant genetic disease, but it's not present at birth clinically. In fact, it's a late onset yet progressive disease. Now, the average age at diagnosis is usually in early teens on the average, but many of these cases actually develop symptoms much earlier, and there's a kind of long diagnostic odyssey before the correct diagnosis has been made. And there are cases, for example, are reported patients as early as 2 years of age. And for example, Sharon kids were diagnosed in the mid-first decade, if I remember correctly. But then there are patients who were not diagnosed until their 20s or 30s. So there's this kind of phenotypic heterogeneity in these patients. And there is also considerable inter and intrafamilial variability in the presentation. In that, in some families, a one organ system, let's say skin, is really severely affected, and they have relatively little eye problems or relatively little vascular problems. While in other families, they may have very severe eye problems and cardiovascular problems, but very little eye problems. So that's known as inter and intrafamilial heterogeneity. So I said sometimes the diagnosis is made as early as in 2 years of age. And that has also been called as a pediatric PXE. Now, a few years ago, we had this 2-year-old little girl, and the doctor noted, pediatricians noted skin changes on the neck. And if you look more carefully, yes, she has kind of very early, kind of these yellowish small papules, which are PXE-like changes. And if you took a biopsy from the neck and did the staining that Dr. Berkowitz was explaining in this case, von Kossa stain, all these black dots are calcium deposition. So she has early changes of PXE in terms of clinical findings on the neck and histopathology findings of calcification of elastic structures in the skin. So we thought this is ABCC6 and went to identify mutations. She did not have ABCC6 mutations. She had mutations in another gene known as ENPP1 gene, and this just shows the kind of techniques, how we found it. And we note that this ENPP1 gene has been previously reported in association of generalized arterial calcification of infancy. We call it GACI. The Europeans call it GACI. Some people call it GACI, whatever. We call it like PXE, it's GACI. And that's another autosomal recessive disorder like PXE, but it's a very severe vascular mineralization. In fact, it's present at birth, often even diagnosed at the early prenatal diagnosis by ultrasound. And that leads to arterial stenosis and intimal hyperplasia and causes myocardial infarcts leading to death in early infancy in about half of these patients. So half of the patients die within the first 6 months. So very severe vascular calcification development. But those who survive may develop PXE-like skin findings. So there's this clinical overlap. Now, actually, if we look at these two conditions, both have vascular mineralization, but PXE has a sort of late onset, less severe vascular mineralization than GACI. PXE shows the skin and eye findings, but not in GACI present so evidently. PXE is late onset, GACI very severe prenatal and perinatal diagnosis. And PXE patients have mostly normal lifespan, while these patients die usually in about half of the cases before 6 months of age. Now, initially, we identified ABCC6 gene causing mutations causing PXE, while ENPP1 causes ABCC6. But more recently, we have shown that actually there are PXE patients who have ENPP mutations, and they are GACI patients who have ABCC6 mutations, though this is known as genetic heterogeneity, so that, for example, in this disease, there can be two different genes giving similar kind of clinical findings. Now, I showed you that we studied the mutation detection in 2000. Now there is something of the order of 400 to 500 to 600 different mutations that have been identified. And there are two different databases. One is the publication-based. So there are publications as many as over 400 different types of mutations in PXE patients. And actually there's this ClinVar database, which is just laboratories report, they are not necessarily published. So as many as 600 distinct variants in ABCC6. And if you look, there are different kinds of mutations. There are those MISSENSE mutations, basically a single amino acid substitution in about half of those cases. Stop codons, nonsense mutations make a non-functional shortened protein, and then different kind of consequences of the mutations. So why do we want to know the mutations in PXE? If you have PXE, you have PXE. Why is it important actually to know that there are mutations in ABCC6 or in some cases in ENPP1? Well, it confirms the diagnosis. You can clinically say, yes, this is PXE, but you definitely confirm the diagnosis if you have two ABCC6 mutations. That's kind of diagnostic criteria, as Dr. Berkowitz was explaining. But you can also use the mutation analysis for pre-symptomatic testing. In other words, predict if a child is going to develop PXE in families that have history of these conditions. You can also do carrier detection, look at for those heterozygous carriers, and then provide genetic counseling. What is the risk of recurrence in the family? And finally, perhaps very importantly, is that there are new allele-specific treatments coming in the realm of personalized medicine. And that means that they work allele-specific, they work only for certain kinds of mutations. So you need to know what gene is affected and what kinds of mutations you have in order for these new treatments which are in the pipeline to work. So therefore, I strongly suggest that you all enter the PXE International Biobank, give a little saliva sample, and we can do the mutation analysis. So this pre-symptomatic diagnosis. Well, this this story started about 12, 13 years ago when Lily and I were in Budapest in a international PXE meeting sponsored by PXE International, organized by our colleagues in Hungary. And one of the Hungarian dermatologists came to talk after my talk and said, well, I have this PXE patient. She's a 13-year-old female, has PXE. How can you confirm that? And also, she has a younger brother. We don't know if this brother is at risk of developing PXE. And here is actually the patient. If you see these PXE-like changes, histopathology of the skin confirms this calcification, and the patient has angioid streaks, so clearly clinically PXE. Now, here's the pedigree, and here's the 13-year-old female affected with PXE. She had an older sister, 17 years old, no evidence of PXE. And here is the 7 or 8-year-old brother, and they were wondering if he is going to develop the PXE. So what we were able to do, we sequenced her ABCC6 gene and identified 2 homozygous ABCC6 mutations. So that confirms that she has the PXE, definitely. The parents were heterozygous carriers. They were clinically normal, but they have 1 copy of the bad gene and 1 copy of the good gene, both in the mother and father. And then we sequenced the younger brother, who was 7 years of age, actually. And we found that the brother was heterozygous carrier of those 2 mutant alleles from the parents, and therefore predicted that the brother is a heterozygous carrier at the age of 7, which is well before the age that we usually, or in average, diagnose on the clinical basis. So we can predict that this brother is not affected with PXE. Now, how about these kind of families where there are 2 PXE women, both married, a clinically unaffected guy, and what is the risk of them having a affected child? Now, in this family, all the 3 females here are unaffected, but we can now predict if this union is going to have a risk of having PXE. In other words, would this man be one of those 150,000 heterozygous carriers walking on the street in the United States, and by chance he is marrying this woman who's affected with PXE? So we can now genotype this man, and in this case we found that he does not have any ABCC6 mutation, so there is no risk for these children to develop a PXE. If he would be a heterozygous carriers, then there would be a risk, actually 1 in 2 them affected. So we can predict what happens to the children by looking into the parents' genotype even before they will be having and contemplating of having children. Now, then coming back to this autosomal recessive inheritance pattern. So you have parents are clinically normal. They have 1 bad copy, the orange copy of the gene and good copy of here, okay? And then the risk of 1 of the 4 children gets 2 bad copies, so is affected by PXE. 2 of them are heterozygous carriers, are clinically unaffected, and 1 is unaffected, perfectly normal DNA. So now comes the question. They have the first affected child, and they don't want to have another affected child in the next pregnancy. So again, the next pregnancy has 1 in 4 chance of having an affected child. So what can they do? Well, they can adopt, and then there's a high likelihood that the child is not having a PXE, or they can do a prenatal testing, a genetic testing from chorionic villus sample, but that's usually done at 10th to 12th week gestation, and that would then involve decisions about termination, and that's usually not a consideration in case of PXE. But what they can do, and we have actually seen requests, is something called pre-implantation genetic diagnosis. So you basically diagnose the embryos even before the pregnancy starts. And that's done in the context of in vitro fertilization. So you allow these embryos grow in the laboratory to 4 cell stage level. You just take very gingerly 1 of those 8 cells, you analyze those embryos, and then those which do not have the mutations in ABCC6, you put those back to the uterus of the woman, the mother, to carry the pregnancy. So they know there's definitely no ABCC6. You can actually test for these embryos for all kinds of diseases. So this is coming, and actually it's basically, this just shows, here is a in live, there is this 8-stage embryo. You use a little kind of sampling pipette. You take 1 of those cells out, you analyze, and now you put those cells that do not have the ABCC6 mutations, put them to the uterus. Now, so the advantages of this pre-implantation genetic diagnosis, it avoids the affected pregnancy. So you are not worried at all whether this your child is going to have a PXE. And the diagnosis takes place before even the pregnancy starts. So their termination is not an issue, which can be more acceptable to certain religious and ethical viewpoints. And in fact, there has been subsexual applications to a number of diseases, as these references attest to all about 10, 20 years ago. There are some issues. Cost of the in vitro fertilization can be relatively high, and some insurance companies in the United States pay, some don't. So you have to, if you contemplate this, that may be a consideration. And then the efficiency of in vitro fertilization. Usually it's about 30% efficiency. So that means that 1 in 3 on the average embryos kind of survive. And if they don't survive, you have to redo the fertilization screening. Now, so for this reason, usually you put 2 or maybe 3 embryos back to the uterus. Now, I happened to be in China in 2011, and there was in TV, Zhongzhou TV, was an kind of news about this pre-implantation genetic diagnosis. And you know, the Chinese are very efficient. So instead of putting 2 embryos, somebody put 8 embryos back to a woman's uterus to get children. And lo and behold, they all got 8 wonderful children, so octuplets. Of course, these do not have the disease that they were tested for, but 8 children, that's a handful, I would say, to anybody. You see, they are very happy. They all look the same, except this poor child is not so happy around being here. But nevertheless, so it is a option to avoid affected pregnancies in families that have risk of having an affected child in subsequent pregnancies. Let me talk about little phenotypic heterogeneity. So clinical observations suggest and show clearly, and PXE can manifest with different degree of severity in the skin and eyes and arterial blood vessels. So we have been postulating that there are genetic modifiers, and it's actually known that there are genes that can modify the severity of the disease, essentially all monogenic disorders. So why not in PXE? So if you have 2 copies of the ABCC6 mutations, but there are the mutations in other genes that could make it more severe disease, that's the question that we recently addressed. And this was a study done in collaboration with PXE International. We looked for modifier genes, focusing on specifically on these genes, which are known to be causing PXE-like disease, skin disease, or calcification. And so we wanted to look into PXE patients, whether they have mutations in these and other genes, whether there's a difference in their kind of severity of the disease compared to patients in the same family who do not have mutations in these genes. So we needed DNA samples from family and clinical data, and PXE International provided us from their biobank DNA samples and the clinical data from their databases. And we used this 29 gene array. Basically, you look 29 different genes. You use this very sophisticated next generation sequencing approach to identify the mutations. And then you look the genotype phenotype correlations. In other words, is there correlation of the severity of the disease and the types of the clinical manifestations with the mutations that we find using the PHENODEX. And the PHENODEX is a kind of clinical severity index that was developed by PXE International a couple of decades ago. So we had the DNA samples from 500 PXE patients, came from PXE International Biobank, and we did this next generation sequencing. And using these sequencing technologies, we identified ABCC6 mutations in 92% of the patients, which is a pretty good yield. And this paper is actually recently accepted for publication in Genetics in Medicine. And this actually shows that we found, yes, in 92% of the patients, we found ABCC6 mutations. Some of the mutations were very common, like this arginine to 1141X top codon counted about 40% of all mutations. But then, interestingly, we found mutations in those other genes in addition to the ABCC6 mutations. So GGZX mutations, SAMD9 mutations, ENPP, GAL-T mutations, etc. So we then focused on these genes because they have a little different genotype. So the questions you ask by PXE patients: do you have bleeding or bruising tendencies? Because regular PXE does not have, but there are these variants where you have modification from the GGX gene, then you can have bleeding tendencies. Do you have very severe kind of redundant sagging skin? Again, goes with these mutations. How about ENP1? Do you have early vascular calcification? Were you born with vascular calcification? Is the diagnosis made at the very early onset? SAMD9 is sort of a localized mineralization, mostly on the sites of trauma, so elbows, knees. Are those the sites where you have mineralization? Not so much on the neck, but mostly on the elbows and knees. And is there any evidence of inflammation, in other words, redness of the skin in the areas that develop the calcification? And then there's this kind of very rare disease called BNT5E gene mutations. And they get very severe vascular calcification, but it starts not until in about 50s or 60s of the affected individuals. But they also have earlier periarticular calcification, so means they have severe calcification in their ligaments in the knees and elbows. So they can have, for example, difficulty in walking at the earlier stage, but no skin findings, only vascular calcification. So you ask those questions, and then you might find mutations in these different genes in addition to ABCC6. Now, there is a question, do you find genotype-phenotype correlation? In other words, do different kinds of mutations give you different organs being affected? PXE International, we did an early analysis as early as 2007, and we had 186 cases with 2 ABCC6 mutations and did not find any significant correlation between the types of mutations and the age of onset or the severity of the disease. There is also a more recent Japanese study, no significant genotype-phenotype correlations, but then there was a pretty large French study published also in 2017. 458 cases from France, and they looked for the types of ABCC6 mutations, and they found that in certain kind of mutations, those loss of function, the nonsense mutations, predispose more to vascular and eye complications, so more severe disease. So the question is, how about our current panel that is now in PRESS 2021? Well, we had initially 401 confirmed cases with ABCC6 mutations, and 280 of them had a complete phenodex information from the PXE international database. And as you know, the phenodex measures the severity of the skin involvement, eyes, gastrointestinal tract, vascular involvement, and cardiovascular involvement. And there's these different severity indexes. So you get assign a number and you add them together, and the higher the number, the more severe the disease. Now, in our case, we found this is so-called radar plot, and not to go into details too much, but we see a little difference there in that the eye problems may be more severe in those patients who have a loss of function mutation, but this was not statistically significant. So it's suggestive, more severe eye problems, but while the gastrointestinal, skin, vascular, and cardiac did not make any difference. So to add to this list, our current panel 2020 showed no statistical correlation between type of mutation and severity of the clinical findings. Now, there's been lots of work over the years. And as I said, the ABCC6 mutations were discovered in 2000. We developed, and others developed, the ABCC6 knockout mouse. They were published 2005. And those knockout mice actually recapitulate the clinical features of human PXE very closely. They get vascular mineralization, they get the eye problems, they get skin calcification, etc. So these mice have served as a very good model to try to develop therapies for PXE. For example, we did a magnesium study for various reasons. We looked into different minerals, and we found that feeding the mice with a diet which is high in magnesium completely prevented the mineralization. So that mouse study then led to a clinical trial, which was published 2019. And Dr. Wayne was alluding to this study, explaining actually that it was a relatively small study, only 40 patients, and 20 of them got the magnesium enrichment, and the 20 did not. So the results were not conclusive. Another observation with our study was that bisphosphonates, such as etidronate, actually can prevent the development of calcification in PXE mice. How about in humans? There was a 2018 a Dutch study which suggested that there is a slow down of progression of PXE, but this study is still under evaluation, extending their initial 1-year study to beyond to 5 years. One of the milestones in PXE research was 2013, showing that there's a reduced levels in plasma inorganic pyrophosphate in PXE patients. And this was done by Dutch people, Koen van de Wettering, who worked in the Dutch Cancer Institute, and he's been now at Jefferson in our PXE International Center the past 5 years coming up. And he is working on development of new drugs for PXE patients. And one of the observations was that there is this reduced PPI level. So we have then we and others have been doing preclinical studies in the mouse models, giving the mice this PPI. Why don't it feed PPI, see if the plasma levels go up? And plasma levels go up indeed. So now there is, for example, a clinical trial looking into the PPI administration to patients with PXE and what complications might be, how often you have to administer how much drug, and does it really work? So these are all examples how the basic development, like developing these mouse models or finding the PPI reduced levels, can result to preclinical observations, which then directly feeds to clinical trials. So it's important to support the basic research with therefore having translational implications and hopefully treatment and cure for patients with PXE in the very near future. So we now know, for example, that PPI, the inorganic pyrophosphate, it in itself has a very complex pathway for its synthesis and its degradation. And there are now these different steps which we can target by different manipulations, inhibitors, small molecules, protein replacements, etc. So now there are already several clinical trials in very early stages or just starting. One is, for example, the oral administration of PPI, as I mentioned, or some derivatives, which may be more efficient than the PPI by itself. I mentioned the administration of etitrogenate. It's a stable analog of this inorganic pyrophosphate, and that study is ongoing. Preliminary data have been submitted, suggesting that perhaps there is something, but it needs to be having a longer follow-up. There are early clinical stages of development of oral administration of this kind of inhibitor of an enzyme known as TNAP, tissue non-specific alkaline phosphatase. And if you inhibit this enzyme, you prevent the degradation of inorganic pyrophosphate in plasma. So you may basically expect to increase the PPI levels and therefore prevent the further calcification in PXE. There is also infusion of a recombinant ENPP1 protein. And this is the protein that is missing those GACI patients, but also it has been shown in the mouse models that ENPP1 protein infusion to mice increases the synthesis of PPI, and that is entering early clinical trials, not only for GACI, but also PXE as well. And then I mentioned there are plans for these allele-specific therapies. So you need to know the types of mutation because it works only on certain patients with certain type of genetic disorders. So get your DNA tested and we know your genotype. So with that all, it's been lots of work. Here are some names that have been working at Jefferson in these projects. These are more recent people, but they all have been working together with Koen van de Wettering and particularly Lily, who runs the laboratory and all the mouse studies. It is a close collaboration. We are thankful to PXE International. Matt, Tamara, Mary, Eliza, Erin, Elizabeth, and Yen have been very helpful to us. And of course, Sharon Terry is a close collaborator for many years. But we particularly want to thank all patients and families, your participation, and we look forward to working with you on these new clinical trials that are coming in the pipeline towards development of treatment and cure for this currently intractable disorder. And then finally, supported by taxpayers, the National Institute of Arthritis and Musculoskeletal and Skin Diseases, part of NIH, and the PXE International. And here is our research group. This was 2000 when the PXE mutations were identified. That this was our cutaneous biology research group. Not all these people are working in PXE. We do some other stuff that's, well, other diseases, mostly heritable diseases. This is 2016, at the time when PXE International Center of Excellence, supported by PXE International, was established at Jefferson. And many of these people, actually most of the people, still work with us on PXE and other genetic diseases. And we keep a very close touch with PXE International. In fact, Mary was just visiting us about 3 weeks ago, last month, here in PXE International Center of Excellence in Research and Clinical Care at the Jefferson Institute of Molecular Medicine. And here is Lily Kuhn. Was good to see Mary and yours truly. So thank you very much for your attention, and I'll be happy to answer questions at the end of this session. So thank you very much.
Great, thanks very much, Jouni. Excellent presentation as usual. Okay, so we have a few minutes for questions before we have our next speaker. Jouni, maybe you could take the slide down so that we can see the people. Great, thanks so much. So, questions. That was a lot to digest, I'm sure. Okay, if we don't have any, that's fine. There will be a panel at the very end after Scott's talk.
Sure, I had a question. It might be a little hard to type given some of the technicalities here.
Sure.
If I understood, this is inherited recessive, correct? You need
Yes.
both. You need both mutations, right?
Yeah,
So,
absolutely.
So what if someone only has one mutation, but they have effectively a lot of the symptoms? What is kind of the current thinking or understanding of a situation like that?
Yeah, so the disease is autosomal recessive, and therefore, by autosomal recessive definition, heterozygous carriers by themselves do not have the disease. So that goes with the disorder. Now, if that patient or this individual has skin findings, has diagnosis, has something suggestive of PXE, I can imagine 3 possibilities. One, that we did not find the other mutation in the other alleles on the ABCC6, and that probably accounts for about 5 to 8% of all cases we cannot find because the complexity of DNA does not allow find some mutations which are at the areas of DNA which are not accessible to the mutation. The other possibility is that there is something called digenic inheritance. So 2 different genes, 2 heterozygous genes, one ABCC6 mutation and other, say, GGCX mutation or ENPP1 mutation. And when you put together, you get PXE-like clinical feature. There is no evidence of digenic inheritance in PXE as yet, but it has been shown in other diseases. And the third possibility is that what we see in the skin is not PXE. Maybe it's something that mimics PXE-like changes, like Dr. Berkovich was explaining some of these elastotic disorders have nothing to do with PXE, no calcification, and no ABCC6 mutation. But those would be interesting families, interesting cases to examine at the genetic level.
Thank you.
Thanks, Jouni. And we'll take the rest of the questions